Swayam Prabha. Digital India. Educated India.
Hello students, in this lecture you will be getting acquainted with various characterization techniques for nanoscale materials, and we will be discussing in details the basic working principles of transmission electron microscope, XRD X-ray diffraction, UV visible spectroscopy and DLS, that is dynamic light scattering. So why do we need to characterize nanomaterials? As you see that characterization is going to link the synthesis and functionality of a nanomaterial. The properties of nanomaterials, as they depend on size, shape and structure. So it is important to characterize the nanomaterials, because this is how you will figure out if a particular material is of a particular size, shape or structure. So you can easily determine the function of it. So characterization, this is how characterization links synthesis to functionality, and it is very essential for reproducibility and quality control. So if you need to know the size, the structure, the shape and the way it interacts with its environment. The shape, size and structure of a nanomaterial and the way it interacts with its environment, you can figure it all with the characterization techniques. So there are various characterization techniques to know each of the property of a nanomaterial, and it is very essential to know these properties, because this is how you can determine the functionality of it, you can validate the functionality of it, and it is also essential for reproducing those experiments again. And so it is very important to characterize nanomaterials. So it is very critical for nanobiotechnology and biomedical applications, because you need to know the functional groups, you need to know how they are interacting. So because if you consider biomedical applications, these things need to be very precise. So let's start with characterization of nanoscale materials. So the common characterization techniques, basically these can be divided into structural and morphological techniques, something that figures out the structure and morphology of a nanomaterial, that is the structural and morphological techniques. They can be the characterization techniques can be divided into all these structural and morphological techniques. So optical and spectroscopic techniques are basically used where optics are used for characterizing the nanomaterial. Then particle size and colloidal stability analysis. Particle size is analyzed and the stability of the nanomaterial in a colloidal solution, that is all analyzed via a particular technique. Then each technique provides a complementary information. So each of your technique is going to provide a complementary information. So let's start with the transmission electron microscopy. So transmission electron microscopy is similar to the light electron microscopy. The difference is that there you project a light beam over the sample. Here you are projecting the electron beam to visualize a sample. So visualize a sample like the detailed to create a detailed image, you are projecting an electron beam on a particle instead of a light beam. You are bombarding it with a high energy electron beam. So transmission electron microscope uses a high energy electron beam that is transmitted through a thin sample. So you need to take a thin sample, because like the surface topographies can only be observed when you are using a thin sample. So your transmission electron microscope basically uses a high energy electron beam that is transmitted through a thin sample to project its high definition image on the screen. Now your electrons, the electrons interact with atoms to form an image. So these electrons that fall on the sample, they interact with the atoms of the sample to form an image. So the high electron beam that is falling on your thin sample, they will interact with the atoms in this sample to form a clear, well defined image of this sample. Then It provides a direct visualization of nanoparticles. So, it is providing you with a direct visualization of nanoparticles. What happens is that when you see an image, you see its reflection, but in this case, you are getting a direct visualization of nanoparticles as the electron beam is passing through that sample. So, there is also high spatial resolution down to atomic scale. So, as you can see, the electrons are interacting with the atoms of the sample. So, there is a high spatial resolution down to the atomic scale. So, down to the atomic scale, so there is a high spatial resolution. So, the image is a very high definition image, a clear image. You can obtain that from transmission electron microscopy. That is a picture of a transmission electron microscope. This is connected with a system, and that is the tunnel where the electron beam will come through, and that is the screen where your image will be projected. So, this is how a transmission electron microscope actually looks like. So, that is what the transmission electron microscope looks from inside. So, you can see this is what it looks from outside, this is how the instrument looks like, and this is what it looks like from inside. Through this we will see how it actually works. So, that is the electron source from where the electron beams come. So, an electron beam is focused from an electron source through anode and all the way through condenser lenses. It is condensed from the condenser aperture, it is condensed, and then to the stem scan coils, then through the objective lens to the sample. So, from here to here, from all the way to the objective lens to the sample, and that is the X-ray detector. Okay. So, electron beam is passing through the sample to the objective aperture, to the selected area aperture, then through the projector lenses, then to the detectors, and from here to the viewing screen, which is CCD camera or photographic plates. So, the viewing screen is over here. So, you see the sample is your sample is basically over here, electron beam is passing through it and going to fall on this screen, and it will create a very precise picture of your sample over here. So, and this will go from magnetic prism to the CCD camera or ELS detector. So, that is how you achieve that on a screen. So, now these are nanoparticles, that is the image of copper oxide nanoparticles, and observed through a transmission electron microscope. So, this is a real time image of a copper oxide nanoparticles, that is an experimental image of a real image of a copper oxide nanoparticles that is seen through transmission electron microscopy. And as you can see, the morphology of copper oxide nanoparticles is so clear, and so you can also see the frequency, size distribution and size of the nanoparticles. You can determine all these with the help of electron microscope. So, that is such a clear and well defined image that is obtained from transmission electron microscope. So, that is an image of copper oxide nanoparticles. That is TEM characterization, that is transmission electron microscopy characterization. So, this characterization technique has shown you the shape, the size and the interaction and the frequency of size, okay, frequency of size distribution of copper oxide nanoparticles. That is the scale bar of 50 nanometer. So, you can compare the size of your nanoparticles. So, what information did we get from the TEM analysis? We got the information about particle size and shape, how small the particle was. We can compare that with other particles, and the shape of the particle, whether it is circular, star shaped, square or triangle, whatever. So, we are getting the information about particle size and shape. Then morphology and aggregation state. Then we are getting the surface, the morphology, topology, whatever it is. So, we saw that the copper oxide nanoparticles in our previous image, it is circular, spherical. So, that is the morphology and aggregation state. So, whether they are closely aggregated, so whether they are closely clustered or they are spread out, all these information you are getting from this TEM image. So, you are getting an information about morphology and aggregation state. Then crystallinity and lattice fringes, you are also getting an information about its crystal lattices and lattice fringes, and core shell and internal structures also. So, that is for the bigger nanoparticles. The core shell and internal structures can also be seen with that. transmission electron microscopy analysis. Then there comes X-ray diffraction technique, that is XRD. So, XRD is based on the diffraction of X-rays by crystal planes. So, you take a crystal lattice and you make an X-ray fall on that, and the light, your X-ray beam is diffracted based on the Bragg's law. So, there is a constructive interference which is described by Bragg's law, and it is used to identify crystal lattice structure and phase. And peak broad, what happens is that when you observe on the screen, the peak broadening will indicate the nanoscale size. So, you will observe as the light will be diffracted by the X-ray and recorded on the screen, you will see that certain peaks will be obtained, and what will peak broadening will indicate the nanoscale size. So, that is a picture of how exactly works. That is an X-ray tube, a beta filter, solar slits, divergence slits, and incident beam. So, the incident beam, incident X-ray beam is coming from this X-ray tube all the way down to your sample, and then getting diffracted as a diffracted beam through anti-scatter slits, solar slits, and receiving slits, and all the way to detector. So, here your diffracted beam is getting detected. So, this is calculated via your Bragg's law, and this is the angle at which the incident beam was falling, which is theta, and this is 2 theta, the diffraction, the angle from here, and the entire angle, this is 2 theta. So, it is getting diffracted like this. So, n lambda is equals to 2d sin theta. This is how it is calculated, and Bragg's law determines the entire process of XRD, and your image is formed on this detector. So, this is how your peaks are obtained on different voltages, that is how your peaks are obtained. Peak position 2 theta for copper. These are the peaks obtained, and the peak broadening will tell you about the nanoscale size. That is Bragg's law, which is n lambda is equals to 2d sin theta. This Bragg's law determines this entire process is determined by Bragg's law. So, an incident beam is basically falling on, that is lambda, the wavelength that is falling on incident X-ray beam, that is falling on your atomic structure, lattice, atomic crystal on the atoms of the crystal lattice of your sample, that is the atoms of the crystal lattice of your sample. So, your incident beam is falling in this and a scattered beam, and then falling on another layer and then scattered beam. So, this is how that is the atomic difference between the atomic planes. D is basically the difference between the atomic planes. So, you have different atomic planes, and D is the distance between them. So, that is how you calculate, and that is the principle of X-ray diffraction. The principle is entirely governed by Bragg's law. That is a scattered beam when X-ray is diffracted like this. So, that is theta, that is the distance between the atomic planes, and the principle of X-ray diffraction depends on Bragg's law. Information from XRD. So, what XRD determines is the crystalline phase identification, the average crystallite size estimation, the degree of crystallinity, and the lattice parameters and strain effects. So, as you can see, the X-ray beam is getting diffracted from the different atoms of the lattice. So, it also determines the degree of crystallinity and lattice parameters and strain effects on the crystal. Now, UV visible spectroscopy, this is the basically the first form of characterization, that this is the first type of characterization that we usually use to know at what wavelength, basically what wavelength of light is capable of is being absorbed by your material and absorbed and emitted by your material. So, you determine that via UV visible spectroscopy, the ultraviolet visible spectroscopy. So, this measures the absorption of UV or visible light. Electronic transitions within this actually measures the electronic transitions within nanomaterials. This actually predicts the electronic transitions within nanomaterials, and it is highly sensitive to shape and size, and it is widely used for metal and semiconductor nanoparticles. So that is UV visible spectroscopy. So what is the UV visible analysis of nanomaterials? So this depends on the surface plasmon resonance in metal nanoparticles. As I have already discussed, the collective oscillation of electrons on a material when light falls on it, that is surface plasmon resonance, and which becomes highly localized in metal nanoparticles, which is called as localized surface plasmon resonance. So it depends on the incident light, whatever light falls on it, and which is able to the particular light frequency, that is able to the frequency of the wavelength when it matches with the collective oscillation frequency of the electron. So that energy is absorbed by the electron. Okay. So when the frequency of the incident light, so that is the surface of your material, these are the electrons, and when light falls of a particular wavelength, so there is the oscillation, there is oscillation of these electron, and when the oscillation frequency of these electrons matches the wavelength frequency of the incident light, then a resonance occurs, a collective resonance occurs between this, and light gets absorbed by these particles, these electrons. So light is absorbed, and a particular wavelength of light is absorbed by the nanomaterial. So that is known as surface plasmon resonance. And band gap estimation in semiconductor, you can this can also estimate the band gap estimation in the semiconductor nanomaterials. So UV visible analysis is basically depends on the surface plasmon resonance in metal nanoparticles, and you are monitoring nanoparticle formation and stability. This is a non-destructive and rapid technique. This also estimates the band gap in semiconductor nanomaterials. So that is a detailed figure for the UV visible characterization of a nanomaterial. So what happens is that a particular, when light falls on this, only a particular wavelength is this is a sample, that is a reference. So when this is your sample, your nanomaterial is capable of absorbing only a light of a particular wavelength and emitting that particular wavelength light. It cannot absorb every light of the spectrum, every wavelength of the spectrum. So what happens is that, so that is your light source. So you imagine that, see this is the picture of a spectroscope, okay, spectrophotometer. This is the spectrophotometer, and what happens is that that's the entire setup that is inside this instrument. So this is the entire setup that is inside this instrument, and this is the screen, okay. So there is a light source inside, you switch on the spectrophotometer and you put a reference over here, that is the box for putting a reference and sample. So this is basically this portion, that is the box. You open this lid and you put a reference and you put a sample over here. Reference is basically the solvent that you have used, and or water. You either take water because that is a universal solvent, or you take the solvent that you have used to make your nanoparticle suspension, and then that is your sample. So you put your sample inside this, and what happens is that you switch it on, and there is a light source inside. So it goes on the mirror, reflected like this on a reflector, and goes through a slit, and then there is a monochromator. So whatever wavelength you first scan, whatever wavelength your sample requires to pass through it, like whatever wavelength your sample is capable of absorbing, that particular light will, so that is the monochromator, that particular wavelength of light will pass through it, and the beam slitter and reference, and through the sample, and go from here to here, the detector, and then on the screen, data processing, and this is how the peaks are formed. So whatever wavelength of light this particle is going to absorb, the particular peaks will be formed at that particular wavelength. So particular peaks will be formed for particular wavelengths, and that is how you will get a wavelength scan of this sample, or you can even quantify for different concentrations. If you take different concentrations of your sample, you can even quantify what on a particular wavelength of light, how much of the light is absorbed and emitted through the sample. And this works on the principle of Beer-Lambert's law, that is the wavelength of passing light that is dependent on the concentration of the solution. So that is the principle of UV visible spectrophotometry, that is the incident light and that is the transmitted light, and you subtract that and that is the light that is absorbed by this sample. Dynamic light scattering or DLS. So, what is dynamic light scattering? Dynamic light scattering is based on scattering of laser light by particles in suspension. So, this analyzes Brownian motion of nanoparticles. So, when you put nanoparticles in a suspension, they are randomly moving in and causing Brownian motion, that to and fro Brownian motion that you must have studied what Brownian motion is, the random motion of the particles in a solution. So, based on the scattering of laser, you put a laser light through it and the light will be scattered according to the particles, the motion of the particles. Okay. So, larger the particles, they will be moving very slowly, and smaller the particles, they will be moving faster. So, the light falling onto it and scattering away will help you determine the exact thing based on the scattering of laser light by particles in suspension. So, the scattering of laser light. So, in a suspension, the particles are moving randomly, they are doing to and fro motion, and larger the particle, slower will be the motion of the particle, and the light falling on it will be scattered accordingly. So, larger the particle, slower will be the motion, or smaller the particle and faster motion, and the light falls on it and will be scattered accordingly, and it will be analyzed as a hydrodynamic diameter is calculated, and the data is analyzed. Then it is best suited for colloidal system. As it depends on Brownian motion, it is best suited for colloidal systems. That is dynamic light scattering. So, that is a laser light being focused on these particles. The particles are larger, they are making to and fro motion. So, larger motion means larger particle means that the motion is very slow, and the smaller the particles, they have more space and they will be moving faster. So, laser light is falling on this, and then intensity of light. So, that is the intensity of light scattered. So, that is the laser light falling on this and the intensity of light scattered, and this graph is created and plotted as intensity versus time graph. So, this is the smaller particles, these are moving faster. So, the laser light falls in this, and a particular peaks are created on this intensity time data, and you can see the difference in the peaks, that the larger the particles, slower they will move, and they will create broader peaks, and then smaller particles, faster movement, and they will create narrow peaks. So, this is how you determine the size of the particles also, and you also calculate the hydrodynamic radius, and that is also the same thing. That is the emission fiber, that the reception fiber, laser light is sent through this and scattering light goes out through this, and these are the particles. So, what is the information obtained from DLS? Dynamic light scattering will provide information on the average particle size in a solution. So, it can provide you an information on the average particle size in a solution. As I have already told you, whether larger size or small size particles, it will give you an information on that. In a solution, there may be smaller particles, there may be larger particles, so it will give you an average particle size. It can also give you a particle size distribution. It will give you a particle size distribution, and then aggregation and dispersion behavior. It also tells you about aggregation and dispersion behavior. If these are well spaced, how fast they are moving. If they are aggregated, how fast they are moving, and you obtain information on that based on that. And then colloidal stability of materials. So, as it measures things from as the nanoparticles are moving in Brownian motion, and dynamic light scattering is measuring that. So, it can also measure the colloidal stability of nanomaterials. What is zeta potential of nanoparticles? These can also be measured via the same instrument from the dynamic light scattering. So, how can it be defined? The definition is this: the potential difference between the nanoparticle surface and the bulk liquid. So, pay attention, the potential difference between the nanoparticle surface and the bulk liquid. So, when there is a nanoparticle suspension, and there is a potential difference between the nanoparticle surface and the bulk liquid. It is determined by the immobile fluid layer and the mobile layer of atoms. It is determined by the immobile fluid layer and the mobile layer of ions. So, stability indicator, it is a large zeta potential means it is either positive or negative. It can be either positive or negative, and a large zeta potential signifies strong electrostatic repulsion. Okay. So, when there is a large zeta potential, which means that there is a strong electrostatic repulsion preventing particles from clumping or flocculation or aggregation. So, there is a large zeta potential, which means there is a strong electrostatic repulsion between particles which prevents them from clumping, flocculation or aggregation. Then thresholds, generally plus minus 30 millivolts indicates a good stability. So, there is a threshold for stability. This indicates that there is a good stability, while values between minus 30 millivolts to plus 30 millivolts suggest potential instability. So, this voltage will help you to determine the stability of the nanomaterial. So, factors influencing it: pH, ionic strength, salt concentration, nanoparticle, material, size, shape and surface modifications. All these factors also influence the zeta potential of the nanoparticle. How it actually works? Now, see the working of it. Nanoparticles develop a surface charge in a liquid medium. So, there is an electrical double layer that forms with oppositely charged ions attracting to the surface, which is the Stern layer, and a diffuse layer of ions further out. So, when you put nanoparticles in a liquid medium, an electrical double layer is formed on its surface with oppositely charged ions attracting to the surface, Stern layer, and a diffuse layer of ions furthering out. So, it is like if your nanoparticle is that's your nanoparticle in your medium, it will be developing a certain surface charge, let us say negative charge. It is developing a negative charge. Okay. So, oppositely charged ions will come attract to the surface and form a Stern layer. So, some oppositely charged particles will come and form a Stern layer around this, and an electrical double layer is being formed. Okay. And a diffuse layer of ions is furthering out. Some diffuse layer of ions is furthering out. So, there is a slipping plane, that is when a particle moves, the liquid at the shear or slipping plane moves with it, while the bulk liquid remains behind. So, imagine when this particle is moving, the liquid at the shear or slipping plane moves with it. Okay, this liquid will be moving with it, but the bulk liquid that remains behind, the zeta potential is the charge at this plane. If this moves, the charge that is left behind is the zeta potential at this plane. Okay. So, that is the picture of it. So, that is the surface charge. Let us say this is that is the surface charge on your nanoparticle, that is positively charged, and then it will start attracting oppositely charged particles on it. That will be the Stern layer, and then that is the slipping plane, and that is the surface charge. Okay. So, that will help you figure out the zeta potential of the particle. That is the surface potential, that is the Stern potential, and that is the zeta potential, which measures the potential at the slipping plane. Okay. If this moves, the charge over here, the potential over here will help you determine the zeta potential. That is the distance from the particle surface on the x axis, and that is the potential. So, it can also what does zeta potential help you determine? It helps you determine the nanofluid stability. It determines how long nanofluids remain dispersed. Then drug delivery, it affects interaction with cell membranes and mucus, influencing drug transport. Then material science, it predicts behavior in coatings, cosmetics and electronics. So, it is widely used. The zeta potential is a very important characterization technique. Then measurement, measured using instruments that apply an electric field and track particle movement. So, electrophoretic light scattering or even dynamic light scattering, these instruments can be used to measured using instruments that apply an electric field and tracking particle movements. So, whatever instrument that applies an electric field and track the movement of the particle is involved in measuring the zeta potential. Now, FTIR, that is the Fourier transform infrared analysis of nanoparticle. FTIR analysis of nanoparticles identifies their surface chemistry or functional groups and molecular interactions that are crucial for understanding the stabilization, capping agents, drug loading and biological interactions in the fields from medicine to environmental science. So, by detecting molecular vibrations, FTIR confirms the nanoparticle synthesis, reveals biomolecular bonding, monitors pollution degradation and distinguishes between intact stressed cells and providing detailed insights beyond traditional spectroscopy. So, your FTIR analyzes the surface chemistry of your nanomaterial, like whatever functional groups are present, and that will determine your synthesis methods, because the functional groups depend on the methods of synthesis also, and molecular interactions can be determined through that, like what bonding is present, CO bonding, NH bonding. So, whatever molecular bondings are there, you can figure it out from the FTIR analysis of that material. Then it is crucial for understanding the stabilization and capping agents, whatever capping agents you have been using. So, that also come in the FTIR analysis. Then the biological interactions can also be determined. So, FTIR confirms the nanoparticle synthesis methods also, revealing biomolecular bonding, monitors other properties. Okay. So, what this FTIR data reveals? FTIR data will reveal functional groups, like you will be getting peaks that correspond to specific bonds. So, you get some specific peaks in your FTIR data, like so that is your FTIR analysis. There is a beam splitter, and this is a sample, and this is a sample, and that is the IR source that is going on the beam splitter, moving mirror, stationary mirror, and going through the sample to the IR detector and creating a spectrum. So, that is your sample over here, you put your sample over here. So, that is the absorbance versus wave number, and these are the peaks. That is the absorbance versus wave number plot, and these are all the peaks that you obtain. So, those peaks correspond to specific bonds, that is OH bond stretch at this wave number, this particular wave number 3400, then CH stretch at this particular wave number. Metal oxygen and metallic oxides, like there can be many functional groups in that with a particular bond stretch. Okay. So, that wave number and the peaks determine the particular bond stretch, and that is how you know what functional groups are present in your material. So, your FTIR data will reveal what functional groups are present in your material by analyzing the specific bonds and the correlated peaks at particular wave number. So, molecular changes, FTIR also reveals molecular changes. Shifts in peak positions or appearance and disappearance of peaks indicate chemical bonding or structural changes. So, let us say you are analyzing protein denaturation or DNA binding. So, you will be assessing before and after sample. So, you will be assessing shifts in the peak positions or appearance or disappearance of the peaks. So, say let us say some OH bond stretch was present in a particular data in a particular material, and then you put another material and that peak goes missing. So, you know that this particular bond is present no more. So, you will be knowing that there are shifts in the peak positions and their appearance and disappearance of peaks, which will indicate that this bond has been broken or this new bond has been created. So, that will determine the molecular changes that are happening in your material. So, this is highly sensitive to surface functional groups and adsorbed molecules. So, this FTIR data will be important as it is highly sensitive to the surface functional groups and adsorbed molecules. Example is titanium dioxide nanoparticles like TiO2, and FTIR, you put this for FTIR analysis and you will be getting peaks on 3400 wave number, that is broad peak for surface adsorbed water. So, you will be getting a broad peak. See, you are putting this on the sample plate and analyzing its data, and you will be getting this on screen, the wave number, a peak at this wave number, and you will obtain a broad peak for surface absorbed water hydroxyl groups. You will get that OH bond. So, you will know that at this particular wave number this peak comes, then your material is this. And then at 1600 you will be getting HOH bending from water. At 450 to 800 you will be getting strong peaks, which indicates TiO bonds. So, this is the specific for titanium dioxide nanoparticles. So, this 450 to 800, this is specific for this. So, this peak you will be obtaining, you will be knowing that this TiO2 is present in your Atomic force microscopy or AFM is a powerful technique that characterizes surfaces by scanning a sharp tip across them, creating a high resolution 3D topographic maps. So what it does is that it characterizes surfaces by scanning a sharp tip across them, which creates a high resolution 3D image of it. Then it measures various nanoscale properties like roughness, adhesion, mechanical properties, stiffness, modulus, these are the mechanical properties, and even electrical magnetic characteristics by detecting forces between the tip and the sample in different operating modes like contact, tapping or non-contact. I will be telling these in detail in my next slide. It is versatile for polymers, semiconductors and biological samples, offering unparalleled detail without requiring conductive samples or electron beams. So that is atomic force microscopy for you. So that's the basic working principle. So there's a probe, a sharp tip of around 10 to 20 nanometer is placed on a flexible cantilever and that scans the surface of your sample. There's a probe in this instrument which is a sharp tip that is on a flexible cantilever and it scans the surface. Then there are tip sample forces, that is the tip comes in contact with your sample and they can be an attractive force with the tip and the sample, or they can be a repulsive force between the tip and the sample, and that can cause your cantilever to deflect or to oscillate. Then what happens is the laser, there is a detection tool. So a laser reflects off that cantilever onto a photodiode and that detects the tiny movements. So the tiny movement of the cantilever, that is detected by when the laser light reflects off that cantilever onto a photodiode, and then imaging. So when the light reflects back, it goes on to a monitor and a feedback loop adjusts the tip's height to maintain the constant force, and from there it traces the surface contour to build a 3D image of your sample. So it's like this. So that's your cantilever and tip, and here's your sample. That's your sample with some surface topographies. So that's your cantilever and that's your fine tip, very fine needle. So what happens is that there may be some interaction between this tip and your sample. That interaction can be either attractive or repulsive. So what happens is that this cantilever will oscillate or deflect, and this light, as it falls on this, so accordingly that light will be reflected on the photodiode and that will go to the detector, and accordingly a 3D image will be created. So that is how it works. And this is the instrument. This is your atomic force microscopy instrument, and that's your monitor where you see the image. So here you put your sample and it is analyzed over here, and here you get your 3D image. So let's see the basic working principle again. So there's a probe, that was the sharp tip, that the needle is 10 to 20 nanometer and that is placed on a flexible cantilever and that scans the surface. Then forces, the tip sample forces, these are either attractive or repulsive and they cause the cantilever to deflect or to oscillate. Then detection, a laser reflects off the cantilever onto a photodiode detecting these tiny movements. Then what happens? Imaging, a feedback loop adjusts the tip's height on the z-axis to maintain the constant force, and this traces the surface contour of your sample to build a 3D image of your sample. Then what does this characterization reveal about your sample? It is going to reveal the topography and morphology of your sample. It is going to generate 3D images that reveals the surface roughness. So on the surface, whatever topography is, it will be measured by atomic force microscopy. Then mechanical properties, it measures the local stiffness. It measures the local stiffness, elasticity, adhesion and viscoelasticity. So it measures the mechanical properties. Then electrical properties, it detects conductivity and surface potential. So AFM also detects the conductivity and surface potential. Then magnetic properties, it measures the magnetic force variations. It also measures the magnetic force variations. Then chemical analysis, so it can be combined with spectroscopy for chemical mapping also. So the chemical analysis can also be done through atomic force microscopy. Then in situ studies, this allows real-time observation of dynamic processes like polymer crystallization, melting or deformation under stress. So, there can be in situ studies also where it allows real-time observation of dynamic processes like polymer crystallization, melting or deformation under stress. So, that is your entire unit and that is your instrument. This is how it actually looks like. Now, that's a real-time AFM image of synthesized silver nanoparticles with this extract, Z-official extract. This AFM image is of the synthesized silver nanoparticles. So, you can see there is a big 3D image of your silver nanoparticles. These are your particles, these yellow things are your particles, and that is a 3D image. Then that is a cross section of the representative area from this image. So, that is an experimental image of that's the AFM image of synthesized silver nanoparticles. Then X-ray photoelectron spectroscopy, that is XPS for you, X-ray photoelectron spectroscopy. This is a surface sensitive technique that uses X-rays to eject electrons from a material surface, allowing for determination of its elemental composition, chemical state and electronic structure from the top few nanometers. So, that is also a topographical thing. So, what is happening is that basically this technique is a surface sensitive technique that uses X-rays to eject electrons from a material surface. X-rays are going to fall on the material and electrons are going to be ejected from that material surface. This allows for the determination of its elemental composition, chemical state and electronic structure from the surface, from top few nanometers. Okay. So, it works by measuring the, how does it do so? It works by measuring the kinetic energy of these photoelectrons. So, after the X-ray falls on the material, some electrons jump and you measure the kinetic energy of these photoelectrons, and this provides a detailed information on the elements present, their oxidation states and their bonding environments, this makes it crucial for material science, catalysis and corrosion studies. So, it does so by measuring kinetic energy of these photoelectrons, and this provides a detailed information on the elements present, their oxidation state and their bonding environments. So, what does XPS reveal? It reveals the elemental composition of your sample. It identifies the elements that are present in your sample. Except this, or it identifies all the elements that are present in your sample by their unique binding energies. Okay. So, your elements have unique binding energies and your XPS identifies your sample like that. It identifies the elements present by their unique binding energies. Then quantitative analysis, it determines the relative amounts of each element. So, what does it do? It makes a quantitative analysis. It determines the relative amounts, that is the relative concentration of each element present in your sample. Then chemical state or oxidation state, it distinguishes between different chemical forms of an element, that is sulfate versus sulfide. So, it distinguishes between the different chemical forms of an element due to subtle shifts in binding energy. So, it is hard to make a difference between sulfate and sulfide, but what does it do? It distinguishes between these different chemical forms of an element. It does so by analyzing the very subtle shifts that happen in the binding energy of each element. So, electronic structure, this XPS provides insights into the material's electronic environment. What is the working principle of XPS? First, irradiation will happen. A sample surface is basically bombarded with monochromatic X-rays. So, your sample surface is bombarded with monochromatic X-rays. Then what happens? Electron ejection. What will these X-rays do? The X-rays will fall on your sample and this will cause core level electrons to be ejected from the atoms. Okay. So, what will happen is that from the core your electrons are going to be ejected from the atoms, that is a photoelectric effect. Electrons will cause core level electrons to the X-rays will cause core level electrons to be ejected from the atoms. So, energy measurement, the spectrometer will measure the kinetic energy of these emitted photoelectrons. So, X-ray is falling on your sample, it is causing the core level electrons to be ejected from the atoms, which is the photoelectric effect, and then what is your spectrometer doing? It is measuring the kinetic energy of these emitted photoelectrons that are jumping. So, binding energy calculation, the binding energy is calculated using the X-ray energy, E photon and the measured kinetic energy. Binding energy is calculated. It is calculated by using the X-ray energy, E photon and the measured kinetic energy. So, that is the working principle of your X-ray, photon, electron spectroscopy. So, what is happening is, photons are coming from here, that is the X-ray gun, and the X-rays are coming from here and it is going on to your sample, and from the surface the X-rays excite the atoms and electrons jump from here, and electrons jump from here, and you calculate the kinetic energy of these electrons. So, you measure the kinetic energy of these electrons and then you calculate your E binding, your binding energy from this formula. So, that is E binding is E photon minus E kinetic plus, so that is a constant, that is a spectroscopic constant. So, what is happening is E binding your binding energy, and E photon, what is E photon? That is the energy of your X-rays. So, and E kinetic, what is that? That is the energy of your, that is the kinetic energy of the electrons that are ejected after your sample is bombarded with these X-rays. So, X-rays are in and electrons come out inside the vacuum when the atom gets excited. So, that is how you calculate the binding energy of the element that is present in your sample. So, that is how you know what elements are present in your sample. The binding energy of your electron on each energy level, you can see that's copper 2s, 2p1, 2p3. So, in all the that indicates the binding energy of your copper element. So, that is the XPS survey spectrum of pure copper. So, you can see the binding energy of electrons in each energy level of your copper atom. So, these are the top 20 atomic layers of your sample. So, on the surface of your sample, this is happening. The X-ray photons are falling on this and your electrons are ejected from here, and then binding energy of the electrons is calculated accordingly. So, you come to know about the binding energy of the element that is present in your sample. Then, this is your entire instrument, that is the X-ray monochromator, that is the CCTV color microscope monitor, that is your X-ray source, that is dry pumping, and that is for sample cooling. So, this is the entire setup of your XPS instrument. Now, the scanning electron microscopy, that is SEM. Scanning electron microscopy is a powerful imaging technique that scans a sample with a focused beam of electrons to create highly magnified detailed images of its surface. So, you remember transmission electron microscope, that was also similar in that it was scanning a sample with the focused beam of electrons. But here what is happening? Here more detailed images are, you are achieving more detailed images of your sample surface. So, this reveals the topography, morphology and elemental composition by detecting various signals like secondary electrons, backscattered electrons and X-rays generated from the electron sample interaction, offering much higher resolution than optical microscopes. So, what is happening is that you are getting images that are generated from the electron sample interaction. There is an electron beam falling upon your sample and your electron sample interactions are going to generate 3D highly detailed images of topographical images of your sample, which offers much better resolution than optical microscopes in which light is falling on your sample. So, now scanning electron microscopy is a powerful imaging technique that scans a sample with a focused beam of electrons to create highly magnified detailed images of its surface, revealing topography, morphology and elemental composition by detecting various signals like secondary electrons, backscattered electrons and X-rays that are generated from the electron sample interaction, offering much higher resolution than optical microscopes. So, what the scanning electron microscopy reveals? It reveals the surface topography. So, what do you see? You see a detailed 3D like view of the surface texture and features of your sample. You see a detailed 3D like image of your sample. So, you get an idea of the surface texture and topographical features of your sample via scanning electron microscope. So morphology, the shape and size of particles and structures, that is also what you can analyze from this type of characterization. Then composition, elemental makeup of the sample, often enhanced with techniques like energy dispersive X-ray spectroscopy. So often this can also, your scanning electron microscopy can also be enhanced by EDS and EDX, that is energy dispersive X-ray spectroscopy. That is also going to reveal the elemental makeup of the sample. So what you can do, you can enhance your scanning electron microscopy with techniques like EDS and EDX. So that will reveal the elemental makeup of the sample also, along with the topographical and morphological analysis. Okay. So what is the working principle behind scanning electron microscopy? So it is much similar to your transmission electron microscopy, where an electron gun is there and it is going to fall, an electron beam is falling on your sample, and basically the electron gun is generating electrons, and which are focused into the narrow beam by electromagnetic lenses. So your electron beam is falling on your sample. Then raster scan, the beam scans across the sample surface in a raster pattern, like a TV screen. Your beam is basically scanning across the sample. So this is what is different from your transmission electron microscope, where your beam was being transmitted, but here your beam is scanning across the sample surface in a raster pattern. So signal generation, as electrons hit the sample, what do? They interact with atoms, they interact with the atoms in your samples, causing the emission of various signals, primarily secondary electrons for surface detail and backscattered electrons for the composition. So electrons, basically the electrons that are, so as electrons are hitting the sample, they are interacting with the atoms in your sample, that is causing the emission of various signals, that is primary secondary electrons, that means that is for the surface detail, and backscattered electrons, what do they tell? They tell you about the composition of your sample. Then detection and imaging, detectors are present that capture these signals, and a computer correlates the signal intensity with the beam's position to build a real-time high resolution image on a monitor. So then you can see there are detectors that capture these signals, and a computer is going to correlate the signal intensity with the beam's position to build a real-time high resolution image on a monitor. So that's the working principle. That's your electron beam, that's your electron gun, and an electron beam is coming out of it. That's the anode, and it is being focused through the magnetic lenses, then scanning coils, and then basically, so that is your specimen present over here, and there are these scanning coils, and then your electron beam is falling upon your sample. There are some backscattered electrons and there are secondary electrons. So this is going to be detected like this, and this is going to be detected from here, and from here to the TV scanner. So the principle is that is the incident beam of electrons, and then there are backscattered electrons from your sample. Your sample is present over here, and incident beam of electron is falling on it. Then there are secondary electrons, that is backscattered electron, secondary electrons, backscattered electrons, and then X-rays and Auger electrons. So that's your scanning electron microscope in entirety. That is how the instrument looks like. So here's the entire setup. So that is the working principle and setup in your microscope. So that is present over here. You put your sample and then you monitor it over here. So you get the image on this screen. That's a comparison of TEM, XRD, UV visible and DLS. So what does TEM measure? What does TEM analyze? Your transmission electron microscopy analyzes direct imaging at nanoscale. So you just want to take a picture of your nanoparticle, you go for transmission electron microscopy. So you are getting your clear picture of your nanoparticles in your images. So you will be knowing the shape and size and chemistry of that nanomaterial, then of that nanoparticle. Then XRD or X-ray diffraction, that determines the crystal structure and phase analysis of a material. So XRD will be determining the crystal structure, the crystal lattice, that works on the principle of Bragg's law. So UV visible, that will be determining the optical and electronic properties. Okay, so like what particular wavelength of light is being absorbed by your sample, that will be determined by the UV visible spectroscopy. Or DLS, that is particle size in a liquid media. So what is dynamic light scattering doing? It is monitoring the faster and slower movement of the particle, which corresponds to the size of the particle. Like larger particles are moving slowly, smaller particles are moving faster, and the light that falls on it will determine the, and the data that is obtained from it will be determining the particle size of the nanoparticle in the liquid media. So that is what dynamic light scattering will do. So your characterization techniques will ensure precise nanobio interactions. See, if you want to use your nanomaterials for ensuring precise nanobio interactions, you need to know the exact shape, size, and the property of the material that you are using. You are tailoring a nanomaterial for a particular biological process, then you need to ensure the very precise properties of that material. So characterization is very important when you take these interactions into account, because your nanomaterials are going to interact with biological, whatever biological entity you are dealing with, your nanomaterials are going to interact with that. So it is very important to know if your nanomaterial has a particular charge, they have a particular functional group, and like whatever their structure is, whatever their size is, how they move, and also how they interact with the environment. So you know all these information you'll be obtaining through characterization, and that is how you proceed for any biological interaction with a nanomaterial, which is very important. So it ensures a precise, your characterization techniques ensure a precise nanobio interactions. Then characterization of functionalized nanoparticles. As I have already told you, your nanoparticles are functionalized. They can be functionalized with various functional groups. So they can be intentionally functionalized or naturally functionalized. So you need to characterize what kind of functionalization is present on the nanoparticles. Then there is quality control for biomedical applications, and like for biomedical applications you need to do quality control. You need to know what kind of a material it is, you need to characterize that material. And then essential for diagnostics and drug delivery systems. It is very important for diagnostics and drug delivery systems. You need to know the entire structure and function and property of your material before proceeding to diagnostics and drug delivery systems. You cannot be putting just any nanomaterial inside your body for drug delivery or diagnostics. You need to know everything about it, what kind of a material it is, what constitutes that material, how that material moves, and everything. What are the optical properties, the magnetic properties, everything you need to know before proceeding to drug delivery systems and diagnostics. Or even for environment, you need to know if the material is toxic or non-toxic or whatever. You need to know everything about that material before proceeding to any remediation method or any biological biomedical application. You need to know everything about that material that you have synthesized. So characterization techniques are very important. That is the first step after synthesizing. That is the first step before you proceed to any application. So it links the synthesis of your nanomaterial to the functionality of your nanomaterial. Characterization techniques are going to link the synthesis of your nanomaterial to the functionality of your nanomaterial. So it will, summarizing the lecture: transmission electron microscope, XRD, X-ray diffraction, UV visible, and DLS, and also zeta potential FTIR that we have seen are core nanomaterial tools. These are the tools for determining nanoparticle properties, and these are the tools for nanomaterial. So each technique provides a unique information. So every technique, each of these techniques, this provides a picture of your nanoparticle, that provides the atomic crystal lattice information. This provides the optical electronic property. This provides the shape and size of your particle and how it is interacting, and also zeta potential, that was telling you about the charge of your particle, and so this is how, and also FTIR, that was telling you about the functional group present on the surface of your material. So each technique was providing a unique information, and there are also plenty of other techniques that we can study in detail, and they provide unique information about your nanomaterial. Combined analysis. So not just one analysis, you don't need to just characterize your material in one way, you need to get a combined analysis that gives a comprehensive understanding of your nanomaterial. So, you need to understand a synthesized material in its entirety. You are going to make a combined analysis, use various characterization techniques to make a combined analysis and understand it completely, like what kind of a material it is, how it interacts, how it bonds with a particular material. So, you need to know everything about it. Then it is fundamental for nanoscience and nanobiotechnology research. So yes, it is a very important tool because that determines all the applications that you are going to do. So that is the first approach after your synthesis that you do to determine what applications it is suited for. So that's the characterization techniques for you. Thank you.